transfer RNA, messenger RNA, and ribosomal RNA all have different structure and therefore all have different function. RNA polymerase is essential to having transcription occur, and there are also processing or modifications done to the messenger RNA before it leaves the nucleus, like a tail, a cap, and the introns get spliced out, which can also lead to alternative splicing. So our central dogma of molecular biology is that DNA goes to RNA, and we call that process transcription. RNA goes to protein, and we call that process translation. So we have messenger RNA, transfer RNA, and ribosomal RNA, but we also have some small RNA that we'll get into in another time. So we have this flow of genetic information. We store the genes in the DNA in the nucleus of eukaryotes. However, stored information isn't carrying anything out, so the functional message or actual process that can actually do the job is the protein. So again, we have this double-stranded DNA. Transcription is going to lead us to messenger RNA, which is read with triplet codons. And translation at the ribosome is going to lead to the assembly of amino acids, which ultimately leads to proteins. So again, another way of seeing the same thing. In a eukaryote, we have our DNA up here in the nucleus, which leads to the messenger RNA, which is processed, which then leaves the nucleus and finds a ribosome. At that ribosome, which consists of protein and ribosomal RNA, transfer RNA will transfer the amino acids to the messenger RNA, and those amino acids will bond with each other. So each one of the different RNA molecules has a different structure. Again, our messenger RNA, we often think of it as this long strand, and again it has codons which are red, and they match up with the anticodons on the transfer RNA, which has amino acids which will be assembled into proteins. The ribosomal RNA has these different subunits, but the ribosome itself does consist of ribosomal RNA. So when we start thinking about transcription, where we take the DNA and get messenger RNA, we need RNA polymerase to make that happen. And again, we have a template strand, and the messenger RNA is assembled by the RNA polymerase a nucleotide at a time. So we bring over one nucleotide at a time. We go DNA to RNA when we're thinking transcription, and again, RNA polymerase is making that happen. Now this process doesn't happen and we only get a single messenger RNA. When this process begins at the promoter region, we could get, as long as that promoter region is still available and there's transcription factors available, we can get thousands and thousands of messenger RNA when a gene is actually turned on. So when we have this other image here, we are looking at reading the original in the 3 to 5 prime direction. So we have double-stranded DNA, but only one strand is actually going to be read, and that is our DNA template strand. So it's the template strand that we're reading in the 3 to 5 direction. The messenger RNA is built in the 5 to 3 direction, because those nucleotides can only get added on to the hydroxyl group on the 3' end. So RNA polymerase is going to move down the template strand and create the messenger RNA. Now the non-template strand is also called the coding strand, this side over here, because when we look at this code, it is essentially the same code as the messenger RNA. So the template strand is read, and again the template strand could be called the non-coding strand or the minus strand or the antisense strand. So there's lots of other names, but we typically stay with the template strand. So once we create that messenger RNA, other things are going to happen to it before we can go to the ribosome. We call this processing. So the processing is going to occur within the nucleus, and we have this pre-messenger RNA and mature messenger RNA. So some of the things that occur here is a GTP cap is put on one end, on the 5' end, and a poly A tail is put on the back side. The caps and tails facilitate export. They protect it from being broken down and assist it in binding into those ribosomes. So the next thing we need to talk about is splicing. So the introns are in the way, so they get cut out by a spliceosome. And again, you can see these little things in here. Those are actually small nuclear RNA that are assembled with other proteins in order to make up a spliceosome and cut out the introns. So the introns are cut out or excised by the spliceosome. So when we look at this, the introns are leaving and we end up having the exons put back together. And it's the exons that exit and actually express the gene when we talk about gene expression. But we can do that in alternative ways. So the same DNA gene can make different messenger RNA transcripts. So when we look at this, the numbers represent different exons. We're going to take the introns out, but we could assemble the introns or the exons in different ways. We could go 1, 2, 3, 4, we could go 1, 2, 3, we could go 1, 3, 4, and each of these is going to then create a different code and a different assembly of amino acids, which could create different functional proteins. So while our genome has 20 to 25,000 genes, because of alternate splicing and all of this editing, we could have over 100,000 different possible transcripts that exist from our own genome. So one more time through, we start with our DNA. Through transcription, we get pre-messenger RNA. We put caps and tails on. We splice out the introns, and we end up with mature messenger RNA, which is then ready to leave the nucleus, go to the cytoplasm, find a ribosome, and be translated into protein. Transfer RNA, messenger RNA, and ribosomal RNA all have different structure, which means they have different function. RNA polymerase is essential to transcription, and processing occurs where we put caps and tails on, and we splice out the introns, which can also lead to alternative splicing.